Hello everyone and welcome to my course that I made and particularly made. This is going to be a virology course and you are now taking class 1. This is my first video that I've made and I hope you guys are going to enjoy. So basically this course is going to be studying virology and how we can look at the morphological sequential structure of viruses and how they function. We're also looking at how they are going to be particularly interacting with various cellular components inside of the cellular factory, and how there are various viruses that come in various morphologies and various types. We are also going to just look at a general aspect of how they function. I hope you guys enjoy. So what is virology? Virology is a branch of science that studies viruses. It's a very vague statement, but it correlates to what the title actually means. But it also deals with the fundamental conceptuality of the morphology, the shape and the form, the interaction with other compositions, the biochemical components, and more. Virology is also very helpful in getting a very in-depth aspect into how viruses function and how we can gain a better understanding of how we can treat them. Another clinical correlation is that virology is used with various clinical environments. They are also used with the amount of substantial mass that a patient should receive. Physicians and microbiologists also study how the patient's genetic information could potentially affect the development of the virus. Basically, what this means is that we're looking at how it's gonna fundamentally, we're gonna look at how a patient's DNA or genetic information can affect how the viruses function on their body. It also exercises immunity. That's particularly the main reason why these physicians and nurse practitioners are going to be injecting them in your epidermal or epithelium in order to go in your bloodstream because it's going to exercise your immunity. Um, Many other things. The structure of a virus. So basically, what is the structure of a virus? Sorry, let me go to this slide. The structure of a virus, normally and generically all viruses are going to have a capsule, the individual DNA. The DNA is going to be in an alienated function and operable infrastructure for what they're going to be implementing. Viruses are going to be standardized in various detrimental measurements. The whole general and fundamental dictation on the hazardous function of the virus depends on the DNA and the particular genomic function. Many viruses are genetically engineered in order to be fundamentally exercising the immunity and the lymphocytes. The anatomical morphology of the virus is shown in the picture after this, but I just want to discuss what this actually means. It's going to be saying how the DNA and RNA inside of a particular virus is going to affect its function on the genetics of a human being. So the anatomy of a virus, we see here, okay, this is going to take kind of long to be discussing, but we see here that, um, so we see various types of viruses that are inclusively sociable inside the manifold. So we see a tobacco virus and an adenovirus. So these viruses are going to have various structures and components that contribute to its function. So we see tobacco, adeno, influenza, and T. even bacterial phage viruses. They have various components that are going to be acting on their fundamental conceptualism and their operable functionality of how they're going to be working. So, we we recognize tobacco, generically everyone, as a very harmful substance that can change our DNA in a very detrimental or very hazardous way. But we can see that the tobacco virus and adenovirus, which are actually, yeah, which, I mean, sorry, I mean the tobacco virus and influenza virus, which are very detrimental and hazardous to the human being, are going to have RNA. But now we see adeno and T even bacterial phage and how they're not as harmful, but they both have DNA, but the tobacco and influenza virus both have RNA. Well, we can infer that RNA is going to be the most effective on how they change and alter the genetic information inside of a human being. So basically, I want to kind of go through all the components over here. So in the tobacco virus, it's like a cylindric, and um, so basically, it has a capsule or capsid. Basically, it's just going to be kind of like a membrane. It's going to block all other components from invading. And then we see the adenovirus, and it has glycoproteins, which is the same thing, but they're basically glycogenic proteins in capsomere, which is going to be associated in the DNA, which is going to function everything. And then the influenza virus also has a glycoprotein in a membranous envelope, which is basically going to do the same thing. RNA capsid is going to function and propagate material, and later we now have the head the head of like so now we're just looking at how there's a spider-like structure, and it has a DNA, has the head, the sheaths, and everything, and those are just going to make it all function together. The DNA and the capsule and the sheaths are going, and the bottom of the sheath DNA is going to come out into the cellular factory. So at the bottom of the spider thing, the DNA will be going out in strings.